So all these different kind of query languages and different ways of doing it, we need to manage those. So we built this, Todd Detweiler and our groups built something called a query manager, where you basically choose which language you want to do. You write your query, you can execute it, you can save it, and then you can reuse it. And that's actually turned out to be very useful. So the idea is we might have experts building these queries, and then other people would be able to use them, and especially when we build graphical interfaces on top of them. So one of the query manager has a few other interesting capabilities, but I'll show you that now. So this is that same query and blown up. This is a very, very simple query. We just want to know the parts of the skull. Basically, that's asking for the parts of the skull. And if we execute that query, we'll get these things: calvaria, neurocranium, viscerocranium. But now we can ask, what are the parts of basically a variable, a parameter. And so this is a saved query that has a parameter, and so if you execute this by itself, nothing will happen. But you can call this through a query execution service. So this is basically a REST service where you basically give the URL, which is actually basically asking for the idea of this saved query, which is 95, and attach onto that an argument, which is skull, and that argument then gets substituted for that placeholder here in the query execution service, and then you just get these results here. So that basically, by giving a single URL with a parameter, you can execute the query. Now you could then take one of these values here, neurocranium, and get the parts of that. So you can basically walk through this thing basically by substituting the argument. Now why is that useful? Now we're getting into what we're going to do with these things. We're getting into image annotation. How can we associate the terms from these ontologies with the images? So the way we are doing it is to access an ontology web service to provide these standardized terms. And so inside a program we built called Anode Image, there's a little static string. This is a Java program which gives you that exact REST query, the host and then prefix, which is exactly the same thing, that query number 95 without the arguments. So when you run this Anode Image program, all you do is just keep concatenating onto this string the arguments and then send it out to the web service and get the results back. And then what you can do is put it into the display. So here's just a display of a skull. Actually, here's just shown as a 2D image, but what you're doing is drawing regions of interest around this skull image and then annotating or labeling those regions of interest with standardized terms from that ontology web service. So, but you do that by just clicking this button here, and it'll bring up this term here. And so this is the same results as I showed you before. You start with the skull. It shows the top level parts, and then if the user then opens up one of these things, just like in Windows Explorer, then it finds the parts of those. You can continue to open these things up, and every time you do it, it just sends out a new query to this query execution service. So that's one way to use ontologies to annotate images. You can basically dynamically access this thing without having to load the whole thing into memory. And the output of that thing, this is our system, which we actually did before the semantic wave came out. Basically, it's telling you where the image, what the image was. It's giving you the coordinates of the region of interest, and it's giving you this thing, which is the name we got from the FMA. Nowadays, though, you would use the full semantic web URI for that. And you would probably have a lot more you want to put in that annotation. So Daniel, again, is doing this thing called annotation imaging markup, AIM, which is a CA Big project, which is much richer, but it still has the same information. It just has a lot more in it for annotating images. So that's one thing I might suggest looking at for annotating images is AIM, because it seems to be becoming a standard. I don't know if there are other standards, but that's the one I've heard of. There may be others. And so that is basically, it produces an XML file, which is associated with the image and shows a lot of the annotations. And Daniel's group has also built something, I guess they've renamed it, but it was called something else now, right, Daniel? What? EPAD, yeah. It was iPad, but we can't use that anymore. So anyway, that's a tool that, as any of you know about the Osirix 3D visualization tool, this is a plugin that attaches to that allows you to put rich annotations on top of that. So that's image annotation in a nutshell, and lots of ways to do it, but that's just some examples. Next problem is image management. A lot of people who are in radiology know about PACS systems, picture archiving communication systems. That's the clinical tools for organizing images, and you'd think, why don't we just use PACS? The trouble with PACS, they're huge. They're not that necessarily easy to customized for exactly what you're looking for. So what a lot of people do is they will get their data, the images out of the PAC system in the so-called DICOM format, which is the exchange format for imaging in radiology, and then they will put them in their own customized data management system. There are many, many examples of that. They're generally customized for specific image types, but very few of them also manage these annotations. There are a few. One of them is our own Digital Anonymous Image Collection Manager. Basically, it's a back-end database web front-end that's keeping track of the not only the images, but also these annotations that I just talked about. And so here's an example of how you might use that. You can't read this, but you're searching for one of these annotation terms, which is the rostrum of the corpus callosum, and then it retrieves all those images that are annotated with that using that annotation tool. And then you can choose one of those and then do things with that annotation. We have something called the Digital Anonymous Interactive Atlases, where you can then have it automatically generate a pin diagram showing all the names and pins going to these outlines. So that's all done automatically. You can interact with it, take quizzes on it, various things like that. There are other image managed systems, as I mentioned. Here's the Digital Anonymous. This is another one Daniel's group's doing, biomedical image metadata management system, right? And then another one that's pretty well known now is called XNAT, which came out of Washington University, Dan Marcus. And this is often used it takes DICOM images, and then you use that to manage them locally. Often these are clinical images that you're wanting to access. But of course, when we're dealing with images, as Lucilla's talked about, in this whole group, it's not just images we want to deal with. We want to deal with other kinds of data management. We have a CTSA, Clinical Translational Science Award, and part of my job there is to help investigators manage their data. Images are one kind of data, but there are other kinds of data they need to deal with if they're doing clinical research. Case report forms. A patient comes in as part of a study. You need to collect some information on that, such as just the blood pressure, some of the signs and symptoms you've observed. So a lot of times people use these electronic data capture systems. This is a REDCap system that came out of Vanderbilt, so we've installed there. Oftentimes when the patient comes in, they might want to draw some blood or collect tissue samples. So we're building, or we actually have built a tissue sample management system. We also are working with genomics and proteomics data that might have been done on these tissue samples. So all these things you might want to integrate with your images, as we've just heard. So then the question is, how do you link these together? So you have maybe an electronic data capture system. You might have a proteomics data. You might have a tissue sample data. You might have images that you got out of your PAC system. You've got the ontology that I was just talking about. So how do you put those together? That's the problem of data integration, the last topic here. And there are lots of ways to do it, which many of you probably know about. Centralized repositories. Problem with that is there's so many different kinds of images. They're changing so much in research, unlike clinical medicine. It's often very hard to just standardize on one schema to put everything into. So a lot of times people have all these other data sources out there, and they suck them all in through data warehouses, which I think you mentioned that somebody's doing, but a lot of people are doing that, and we are doing it too in Seattle. We actually bought the Microsoft Amalga system, which is a tool that lets you do that. But even data warehouses can't access everything because things keep changing so fast, and a lot of national systems are using federated systems like CA-BIG, the BURN project, and again, the Neuroscience Information Framework. Now, one other approach that we've been looking at is sort of a more lightweight approach we call distributed queries, where you send out queries to these sources and then they come back and put them together. And that's something we developed as part of the Human Brain Project. We call it DX Brain, and that's the same picture I showed before of all these different data management systems. And these little arrows represent web services. So the idea is to make them accessible on the web so you can query them. And the only criteria that is necessary is that they have to return some form of XML, which is the data exchange over the web. And an RDF can be serialized as XML, so that counts too. So an ontology can be queried through an ontology web service, but all these other ones can be queried too. In fact, most of them do have web service ways to access them. And then we built a query engine that takes a distributed query, sends it out to these sources, and then returns it, and then a web front end that allows you to formulate a query, and then click these buttons at the bottom. One says XML, you get the results back as a single XML document, or another one is a 3D system, so we're working with the brain, so you can actually display it on a 3D visualizer. So let's just look at how you might use this. This is working with Jess Turner at Mind Research Institute in New Mexico, and also Daniel and Marianne Marton, again, who's here. Marianne's here. And so the question we're asking is, do healthy volunteers versus schizophrenics show any difference in fMRI activation in different regions of the brain? And so what Jess did is she worked with burn. This is a bunch of burn data that was acquired on a large study for schizophrenics versus healthy patients on specific functional imaging protocols. So she grabbed that data from burn, it's just as big, huge tar files essentially, and then processed those to find clusters of activation for both the schizophrenics and the controls. And each one of these clusters, these yellow dots represent the XYZ coordinate of the maximal activation that in that particular region. And these maximal activations can be represented as XYZ coordinates in a normalized space, these so-called MNI space. And then, as I described before, you can send these coordinates down to the Talirac daemon and come back with the Talirac labels for these. So that's the original data that we're looking at, and it's represented, can be just saved out as an XML file. And so we use this distributed DX brain predicate to query across these data here and across the FMA through the ontology web service. And so the question we're asking is, are the areas of distribution different in different regions of the brain? So what we first do, this query first goes out to the FMA through this web service, finds out the parts of a particular region, such as the dorsolateral prefrontal cortex is just one of the regions, finds all those parts. For each one of the parts, looks up the associated Talirac term, and then only retains activations that match one of these Talirac terms. This is an example of a so-called intelligent query where you're using the ontology to help you make a query through the actual image data. And then once you get the result back, you can then display it in 3 dimensions using a tool we have called MindSeer, but you could also use 3D Slicer, which Ron's going to talk about a little bit later. So so this is just kind of one way we're using these things to do queries. And then if you then look at the results, all you have to do is change this one term. Here's dorsolateral prefrontal cortex, here's Brodmann area 6, here's frontal lobe, and then the rest of the query is the same, so it's fairly easy to change it. And then the results come up showing the distribution in different parts of the brain. Brodmann area 6, here's the frontal lobe, here's the dorsolateral prefrontal cortex, and then this is the number that, there's a total of 1741 of these centers were initially in the data, and after the filtering, each one retained only a few of those, and then basically this result showed that there's really no difference in this particular data set and this particular cognitive task. In fact, there are differences that the burn study showed, but this was just kind of a calibration study. Okay, that's all I want to talk about basically. But in summary, what I've talked about is the idea that the idea of organizing images through metadata as opposed to content-based retrieval, which is a very important area also. The use of ontologies to tag the metadata so that you can share them. Then making these ontologies, as we build them, accessible to applications, so the ontology web service and ontology views. And then access those in tools that will allow you to do image annotation, and then image management, and then again accessing the ontology and all these sources to do image integration. So in conclusion, I think it sounds like integration is a major theme of this center, and it's a major theme in general, not just here. NIH is very interested in it. Because there's a need to not redo all these experiments. But in order to do integration, you have to have interoperability. The data have to mean the same thing, whether it's genomic data or image data. And you have to have some kind of way to do this, some kind of architecture to integrate them. And I think that what we're saying here is annotation of image data with ontology terms will facilitate the interoperability. There are multiple possible integration architectures, as I mentioned, but all of these can benefit from the use of ontologies. And then finally, I just want to give credit. These are a lot of the UW people that have been involved, both in our structural informatics group and computer science. And, of course, external collaborators, Daniel and Mariana, I've talked about, Jess Turner from Mind Research Institute, and Natasha Noy and Mark Musin from Stanford, funding from NHLBI, NIBIB, NIMH, and NLM over the years. All right, thank you very much.